Hi, I'm Amanda Horn and this is Emma Scholz, and we've been researching malaria, sickle cell anemia, and the ecosystems of Ethiopia by using geographical correlation maps. All right, so first off, we're going to start by giving you guys a little bit of background on malaria. So malaria, it really dominates in Africa, mostly because of the environment there is very suitable for not only the parasite, but also the mosquito. So what happens is you have a parasite, and the two main types of malaria are Plasmodium falciparum and Plasmodium vivax, and those are the two dominant parasites that make up malaria. So what happens is the parasite develops inside of the mosquito, which in this case is known as the vector. Being a vector means that it does not harm the mosquito in any way. It is just used to transmit the disease from person to person. So it gets inside of the mosquito, and then the mosquito goes and feeds on, say, a person. The parasite goes into the bloodstream, goes into the liver, infecting the liver, and then it reinserts itself back into the bloodstream. Once it's back into the bloodstream, it attaches itself to the red blood cell, gets inside of it, and then multiplies. At the same time it's doing this, it's also giving out proteins, letting the immune system know that it's a healthy cell, even when it's not. So it multiplies it so much that the cell explodes. And without your red blood cells, you're not going to be able to get oxygen throughout your entire body, which means that you will eventually end up dying from lack of oxygen. For sickle cell anemia, it is a genetic disorder, and when you have it, your blood cells sickle, such as this one here, and they do not hold oxygen correctly. They cannot hold oxygen when they're oxygenated. They don't work correctly, and so they cannot transfer oxygen to the rest of your body, and you die from not having oxygen. When sickle cell anemia and malaria are connected in the fact that, for one thing, they're both blood diseases, and for another thing, if you have the allele for sickle cell anemia, which means you don't actually have the disease, but you just have the genetic code for it, when you get malaria in your bloodstream, the cells sickle like this one, and your body doesn't host the malaria. Okay, so the purpose of this project is to find the correlation on a larger scale of sickle cell anemia and malaria, which is previously explained, but we also want to look at the ecosystems that are part of Africa, because that is a main part of hosting for the parasite and making sure that the mosquitoes can live how they're supposed to be living. We chose Ethiopia because there is a variety of data there, not only for sickle cell anemia, but also for malaria. So there was a wide range of data for both of those. We also chose it because it is a severe problem there. In a lot of other places, there might be prevention methods and stuff like that, but in Ethiopia, that is not possible. So it's making this a larger problem for them. We chose to do mapping instead of just using data tables, because mapping, you can notice the correlations a lot easier than if you were just looking at a data table. Also, it is very comprehensive. You can look at it and see a correlation before you even run the numbers or run the statistical analysis on it. For our materials, we had to have computer access because, for one thing, we needed to use the internet, which we used for our research to find our data. And we used Summons and Google Scholar for this. Then we also used the software called ArcGIS, which stands for Geographical Information System. And we used this for our mapping. And then we also used Excel for our correlations. Okay, so for our method, start out with where we got all of this information. So for malaria, we started off with some very broad information. We started off with international data and then broke it down into smaller pieces with those references. So using this international data, we looked at the references, the ones that got progressively smaller. And so then we looked at just the information for Africa from those references. And then from Africa, we saw that it had a reference for Ethiopia, which meant that in Ethiopia, this was a big problem. And we went from Ethiopia as an entire country to the regions and then down to the subregions. So that's how we found the raw data for the Ethiopian subregions. And then to find our sickle cell anemia data, we used the information, the references that we found from our raw data in the malaria cases, and because the article that we found had to do a lot with the
it had to do a lot with the correlation between sickle cell and anemia. So we got to find that information. And we found that information in the form of a geotip. And a geotip is kind of like a picture. Instead of seeing a map with all of these boundaries, it's just pixelated. As you can see on the bottom right corner, it doesn't really have any boundaries for the subregions, and so there are thousands and thousands of pixels just in this one piece of information. We also, in our ecoregions, we found those in form of geotips, so they are very, very specific. They are 5 kilometer by 5 kilometer pixels, so the information from that is very reliable.
For our mapping, we started out with by finding our shapefiles for Ethiopia, which means a shapefile is the actual shape of the country and the regions inside of it, and then behind those shapes, the data is there, but it's not shown. Then we had to layer this with smaller polygons so that we could combine them to match up with our data. This had us going from 567 polygons when we added them or combined them into 87. Then for malaria, we used GIS and we inserted the data from our data tables into the GIS software. And then each year had a different map and different different data. And then we had to add legends and the compass and the scale. And then for sickle cell anemia, we had to overlap the maps. We had our original geotip file, and we had to use the longitude and latitudinal lines to overlay it with our shapefile of Ethiopia. And then we had to clip the map from our entire global scale for sickle cell anemia. We had to clip that down to just Ethiopia so that our data was more specific. And then we had to go from a raster data, which looks like this, down to our vector data, which is just the actual data. And we did this using zonal statistics, which takes about maybe 700 pixels per each area here and turns it into one value. So it very narrows down our data. And then we had to add the necessary map qualities to each map. And we did this also for our ecosystems. Okay, so we used 2 different maps to compare our ecosystem and our sickle cell anemia data to. The first one that we used was the malaria incidence average for 2004 through 2009. All of those years were originally separate, but when we looked at it, it could have been, if we had only used 1 year, it could have offset the data. 1 year the data could have not been found, and so taking the average of all of that, it kind of smooths out the rough edges and allows that data to be more consistent. It wouldn't really affect the data very much because the ecosystem, 5 years is not enough time to change an entire ecosystem. So within that 5-year range, the information is pretty reliable. For our sickle cell anemia map and comparing that to the malaria, we did only use the 2008 through 2009 map because the sickle cell anemia information was just for the year 2009. So it wouldn't make sense to compare, say, 2004 to a 2009 sickle cell anemia map. For our significance and define our correlations, we had to use Excel, and by doing this, we used 3 different tests. The first one was an ANOVA test, which takes the distribution of each of the data from each region, ecoregion, and it looks at it to see how likely it is that that data can be so diverse without being correlated. And then we also did a visual correlation, which is only because we could not find actual data, like we couldn't find data for this, we just had pictures, and we couldn't convert it into the data, so we just had to look at it. And then for the trend line map, we would graph the malaria versus the ecosystem data, and then we'd find the trend line that ran through the points, and then we'd look at the R-squared value. And if the R-squared value was higher than 0.95, then it was a significant correlation. This is our malaria average map, which we use to correlate our ecosystems to. Okay, so these are our first 3 maps that we created for the ecosystem. It's average temperature, but then it's also average temperature suitability for the Plasmodium falciparum and Plasmodium vivax to live. So that those temperatures are just suitable to those 2 types. When we ran this against the malaria for 2004 to 2009, we did not find
any statistically significant information. Like she said, you had to get a 0.95 or higher in order to have statistically significant information, and all three of these ran at a 0.0015 R-squared value.
These are our three next maps that we did. The first one is tree cover, the second one is wetlands, and then the third one is herb percentage, which is just ground cover. These also came up uncorrelated.
These two, the average annual precipitation map and the digital elevation model of Ethiopia by region, both of these experienced no statistically significant correlation there.
This is our map for comparing it to sickle cell anemia, and this came up with an R-Scholz value of 0.0031, which is uncorrelated.
Okay, these are our two visual maps, the ones that we just visually analyzed. And although this is just speculation because there is no actual data that you can see from this, you can see in these triangles, there's a black triangle over here in the right corner, and then up here, there's a dark blue triangle. Those obviously correlate, so there may be a visual correlation between those two. And if you look over here, this is actually just a malaria map. The purple is just the malaria map that's in a different shade, so that it will contrast with the blue. And the blue is where the rivers run, and excluding the right side, the bottom right corner, if you exclude that and you look at a river, it is either bordering or running through a dark purple region.
For our ANOVA test, we had to take the standard deviation, and we looked at that, and then our p-value came up to be 1%, which is in the range of correlation. For our terrestrial ecoregions, it's quite possible that this one matched up and had correlation because they have the same accuracy. If you look at this map and this map, they look up to have about the same amount of data. And then also, if you add things together and you add the precipitation and the temperature and stuff, and then you add them together, you're more likely to get a correlation.
So with our sickle cell anemia map, one of the reasons why that might not have correlated as the way that we thought it would is because those two groups of information are at very different accuracies. Like she said, in one of these polygons, there are 700 bits of information for the sickle cell anemia, but there's only 1 point for the malaria. So the sickle cell anemia could have been so specific that it wouldn't even correlate with the malaria when it was all generalized together. Another reason might be because of the evolutionary maturity. Yes, on a molecular level, this can be proven, but it could have not been, it's not widespread enough yet within this country to be able to see it on a broad scale. Also with the ecosystems, the different accuracies, those are again geotips, but also the civil interference. So after World War II, groups in the world tried to eradicate malaria completely. And so if you look at this bottom right corner, that is closest to the ocean. It was reported that they didn't get to the tropic regions, so the dark green areas. They did not get to the tropic regions when eradicating this malaria. So this bottom right could be skewing our data because malaria has been eradicated in that area.
If we were to continue our research or do it again, we would want to have more specific malaria data, such as this dot map, where each dot represents like 10 people or 10 cases of malaria. We would also want to have maybe target a smaller region and only do like one subregion instead of doing the entire country of Ethiopia.
Okay, so to continue our research, we would look at specifically on a molecular level why or why not these may have correlated. So look at exactly how this parasite reacts to, say, precipitation or vegetation cover. We would also look at other ecosystem factors. Domesticated animals might be a big part of that because that would give another host for the malaria to transfer person to person with. We would also look at that exact civil interference location and see if there's any correlation between that.
For our acknowledgements, we would like to very much thank Amy Dunn and Christopher Krause, who helped us to do our research.
We'd also like to thank our sponsors, Noble Energy, the Frank and Gloria Walsh Foundation, for just allowing us to be here and sponsoring our time here at FSI. Also, Noble Energy did sponsor part of this research, along with the Kinder Morgan Foundation, sponsored the other half of it for our project. But we'd also like to thank our facilitators, the University of Northern Colorado and FSI, as well as the NAST office.